Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cilium. 47 languages. Cilium. SEM micrograph of motile cilia projecting from respiratory epithelium in the trachea. Details. Identifiers. Latin cilium. Mesh D002923. THH 1.00.01.1.01014. FMA 67181. Anatomical terms of microanatomy. Edit on Wikidata. This article is about organelles. For fine hairs on insect wings, see cilium, entomology. Not to be confused with cilium, computing. Not to be confused with cilium. The cilium, pl, cilia. From Latin. Cilium eyelid, in. Medieval Latin and in anatomy, cilium is a short hair-like membrane protrusion from many types of eukaryotic cell. 1, 2. Cilia are absent in bacteria and archaea. The cilium has the shape of a slender, thread-like projection that extends from the surface of the much larger cell body. 2. Eukaryotic flagella. Found on sperm cells. And many protozoans. Have a similar structure to motile cilia. That enables swimming through liquids. They are longer than cilia and have a different undulating motion. 3, 4. There are two major classes of cilia, motile and non-motile cilia, each with two subtypes, giving 4 types in all. 5. A cell will typically have one primary cilium or many motile cilia. 6. The structure of the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Eukaryotic motile cilium. Cilium core, called the axoneme, determines the cilium class. Most motile cilia have a central pair of single microtubules surrounded by 9 pairs of double microtubules called a 9+2 axoneme. Most non-motile cilia have a 9+0 axoneme that lacks the central pair of microtubules, also lacking other associated components that enable motility including the outer and inner dynein arms, and radial spokes. 7. Some motile cilia lack the central pair, and some non-motile cilia have the central pair. Hence the 4 types. 5, 7. Most non-motile cilia, termed primary cilia or sensory cilia, serve solely as sensory organelles. 8, 9. Most vertebrate cell types possess a single non-motile primary cilium, which functions as a cellular antenna. 10, 11. Olfactory neurons possess a great many non-motile cilia. Non-motile cilia that have a central pair of microtubules are the kinocilia present on hair cells. 5. Motile cilia are found in large numbers on respiratory epithelial cells, around 200 cilia per cell, where they function in mucociliary clearance, and also have mechanosensory and chemosensory functions. 12, 13, 14. Motile cilia on ependymal cells move the cerebrospinal fluid through the ventricular system of the brain. Motile cilia are also present in the oviducts, fallopian tubes, of female, therian, mammals, where they function in moving egg cells from the ovary to the uterus. 13, 15. Motile cilia that lack the central pair of microtubules are found in the cells of the Embryonic primitive node, termed nodal cells. These nodal cilia are responsible for the left-right asymmetry of bilaterians. 16. Structure, edit. A cilium is assembled and built from a basal body on the cell surface. From the basal body, the ciliary rootlet forms ahead of the transition plate and transition zone where the earlier microtubule triplets change to the microtubule doublets of the axoneme. Basal body, edit. The foundation of the cilium is the basal body, a term applied to the mother centriole when it is associated with a cilium. Mammalian basal bodies consist of a barrel of 9 triplet microtubules, subdistal appendages and 9 strut-like structures, known as distal appendages, which attach the basal body to the membrane at the base of the cilium. 2 of each of the basal body's triplet microtubules extend during growth of the axoneme to become the doublet microtubules. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Ciliary rootlet, edit. The ciliary rootlet is a cytoskeleton-like structure that originates from the basal body at the proximal end of a cilium. Rootlets are typically 80 to 100 nm in diameter and contain cross-striae distributed at regular intervals of approximately 55 to 70 nm. A prominent component of the rootlet is rootaltin, a coiled coil rootlet protein coded for by the CRARC gene. 17. Transition zone, edit. To achieve its distinct composition, the proximal-most region of the cilium consists of a transition zone, also known as the ciliary gate, that controls the entry and exit of proteins to and from the cilium. 18, 19, 20. At the transition zone, Y-shaped structures connect the ciliary membrane to the underlying axoneme. Control of selective entry into cilia may involve a sieve-like function of transition zone. Inherited defects in components of the transition zone cause ciliopathies, such as Joubert syndrome. Transition zone structure and function is conserved across diverse organisms, including vertebrates, Caenorhabditis elegans, Drosophila melanogaster and Chlamydomonas. Reinhartii. In mammals, disruption of the transition zone reduces the ciliary abundance of membrane-associated ciliary proteins, such as those involved in hedgehog signal transduction, compromising hedgehog-dependent embryonic development of digit number and central nervous system patterning. Axoneme, edit. Inside a cilium is a microtubule-based cytoskeletal core called the axoneme. The axoneme of a primary cilium typically has a ring of 9 outer microtubule doublets, called a 9+0 axoneme, and the axoneme of a motile cilium has, in addition to the 9 outer doublets, 2 central microtubule singlets, called a 9+2 axoneme. This is the same axoneme type of the flagellum. The axoneme in a motile cilium acts as a scaffold for the inner and outer dynein arms that move the cilium, and provides tracks for the microtubule motor proteins of Kinson and dynein. 2, 21, 22. The transport of ciliary components is carried out by intraflagellar transport, IFT, which is similar to the axonal transport in a nerve fiber. Transport is bidirectional and cytoskeletal motor proteins Kinson and dynein transport ciliary components along the microtubule tracks, Kinson in an anterograde movement towards the ciliary tip and dynein in a retrograde movement towards the cell body. The Cilium has its own ciliary membrane enclosed within the surrounding cell membrane. 23. Types, edit. Non-motile cilia, edit. In animals, non-motile primary cilia are found on nearly every type of cell, blood cells being a prominent exception. 2. Most cells only possess one, in contrast to cells with motile cilia, an. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Exception being olfactory sensory neurons, where the odorant receptors are located, which each possess about 10 cilia. Some cell types, such as retinal photoreceptor cells, possess highly specialized primary cilia. 24. Although the primary cilium was discovered in 1898, it was largely ignored for a century and considered a vestigial organelle without important function. 25. 2. Recent findings regarding its physiological roles in chemosensation, signal transduction, and cell growth control, have revealed its importance in cell function. Its importance to human biology has been underscored by the discovery of its role in a diverse group of diseases caused by the dysgenesis or dysfunction of cilia, such as polycystic kidney disease, 26, congenital heart disease, 27, mitral valve prolapse, 28, and retinal degeneration, 29, called ciliopathies. 30. 31. The primary cilium is now known to play an important role in the function of many human organs. 2. 10. Primary cilia on pancreatic beta cells regulate their function and energy metabolism. Cilia deletion can lead to islet dysfunction and type 2 diabetes. 32. Cilia are assembled during the G1 phase and are disassembled before mitosis occurs. 33. 11. Disassembly of cilia requires the action of aurora kinase. 34. The current scientific understanding of primary cilia views them as sensory cellular antennae that coordinate many cellular signaling pathways, sometimes coupling the signaling to ciliary motility or alternatively to cell division and differentiation. 35. The cilium is composed of subdomains, clarification needed, and enclosed by a plasma membrane continuous with the plasma membrane of the cell. For many cilia, the basal body, where the cilium originates, is located within a membrane invagination called the ciliary pocket. The cilium membrane and the basal body microtubules are connected by distal appendages, also called transition fibers. Vesicles carrying molecules for the cilia dock at the distal appendages. Distal to the transition fibers form a transition zone where entry and exit of molecules is regulated to and from the cilia. Some of the signaling with these cilia occur through ligand binding, such as hedgehog signaling. 36. Other forms of signaling include G-protein coupled receptors, including the somatostatin receptor 3 in neurons. 37. Modified non-motile cilia, edit. Kinocilia that are found on hair cells in the inner ear are termed as specialized primary cilia, or modified non-motile cilia. They possess the 9+2 axoneme of the motile cilia but lack the inner dynein arms that give movement. They do move passively following the detection of sound, allowed by the outer dynein arms. 38, 39. Motile cilia, edit. Mammals also have motile cilia or secondary cilia that are usually present on a cell's surface in large numbers, multiciliate, and beat in coordinated metachronal waves. 40. Multiciliated cells are found lining the respiratory tract where they function in mucociliary clearance sweeping mucus. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Illustration depicting motile cilia on respiratory epithelium. Tracheal respiratory epithelium, showing cilia and much smaller microvilli on non-ciliated cells in scanning electron micrograph, containing debris away from the lungs. 13. Each cell in the respiratory epithelium has around 200 motile cilia. 12. In the reproductive tract, smooth muscle contractions help the beating of the cilia in moving the egg cell from the ovary to the uterus. 13. 15. In the ventricles of the brain ciliated ependymal cells circulate the cerebrospinal fluid. The functioning of motile cilia is strongly dependent on the maintenance of optimal levels of periciliary fluid bathing the cilia. Epithelial sodium channels, ENaCs, are specifically expressed along the entire length of cilia in the respiratory tract and fallopian tube or oviduct that apparently serve as sensors to regulate the periciliary fluid. 13. 41. Modified motile cilia, edit. Motile cilia without the central pair of singlets, 9+0, are found in early embryonic development. They are present as nodal cilia on the nodal cells of the primitive node. Nodal cells are responsible for the left-right asymmetry in bilateral animals. 16. While lacking the central apparatus, there are dynein arms present that allow the nodal cilia to move in a spinning fashion. The movement creates a current flow of the extraembryonic fluid across the nodal surface in a leftward direction that initiates the left-right asymmetry in the developing embryo. 12. 42. Motile, multiple, 9+0 cilia are found on the epithelial cells of the choroid plexus. Cilia also can change structure when introduced to hot temperatures and become sharp. They are present in large numbers on each cell and move relatively slowly, making them intermediate between motile and primary cilia. In addition to 9+0 cilia that are mobile, there are also solitary 9+2 cilia that stay immobile found in hair cells. 39. Nodal cilia, edit. Nodal cells have a single cilium called a monocilium. They are present in the very early development of the embryo on the primitive node. There are two areas of the node with different types of nodal cilia. On the central node are motile cilia, and on the peripheral area of the nodal cilia are modified motile. 42. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Scanning electron micrograph of nodal cilia on a mouse embryo. The motile cilia on the central cells rotate to generate the leftward flow of extracellular fluid needed to initiate the left-right asymmetry. 42. Cilia versus flagella, edit. See also, flagellum terminology. The motile cilia on sperm cells and many protozoans enable swimming through liquids and are traditionally referred to as flagella. 3. As these protrusions are structurally identical to motile cilia. Attempts at preserving this terminology include making a distinction by morphology. Flagella are typically longer than ordinary cilia and have a different undulating motion, and by number. 43. Microorganisms, edit. Ciliates are eukaryotic microorganisms that possess motile cilia exclusively and use them for either locomotion or to simply move liquid over their surface. A paramecium, for example, is covered in thousands of cilia that enable its swimming. These motile cilia have been shown to be also sensory. 44. Ciliogenesis, edit. Main article, ciliogenesis. Cilia are formed through the process of ciliogenesis. An early step is docking of the basal body to the growing ciliary membrane, after which the transition zone forms. The building blocks of the ciliary axoneme, such as tubulins, are added at the ciliary tips through a process that depends partly on intraflagellar transport, IFT. 45, 46. Exceptions include Drosophila sperm and Plasmodium. Falciparum flagellar formation, in which cilia assemble in the cytoplasm. 47. At the base of the cilium where it attaches to the cell body is the microtubule organizing center, the basal body. Some basal body proteins as CEP164, ODF2, 48, and CEP170, 49, are required for the formation and the stability of the cilium. In effect, the cilium is a nanomachine composed of perhaps over 600 proteins in molecular complexes, many of which also function independently as nanomachines. Flexible linkers allow the mobile protein domains connected by them to recruit their binding partners and induce long-range allustry via protein domain dynamics. 35. Function, edit. The dynein in the axoneme axonemal dynein forms bridges between neighboring microtubule doublets. When ATP activates the motor domain of dynein, it attempts to walk along the adjoining. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Microtubule doublet. This would force the adjacent doublets to slide over one another if not for the presence of nexin between the microtubule doublets. And thus the force generated by dynein is instead converted into a bending motion. 50, 51. Sensing the extracellular environment, edit. Some primary cilia on epithelial cells in eukaryotes act as cellular antennae, providing chemosensation, thermosensation and mechanosensation of the extracellular environment. 52, 10. These cilia then play a role in mediating specific signaling cues, including soluble factors in the external cell environment, a secretory role in which a soluble protein is released to have an effect downstream of the fluid flow, and mediation of fluid flow if the cilia are motile. 52. Some epithelial cells are ciliated, and they commonly exist as a sheet of polarized cells forming a tube or tubule with cilia projecting into the lumen. This sensory and signaling role puts cilia in a central role for maintaining the local cellular environment and may be why ciliary defects cause such a wide range of human diseases. 31. In the embryo, nodal cilia are used to direct the flow of extracellular fluid. This leftward movement is to generate left-right asymmetry across the midline of the embryo. Central cilia coordinate their rotational beating while the immotile cilia on the sides sense the direction of the flow. 42, 53, 54. Studies in mice suggest a biophysical mechanism by which the direction of flow is sensed. 55. Axociliary synapse, edit. With axociliary synapses, there is communication between serotonergic axons and primary cilia of CA1 pyramidal neurons that alters the neuron's epigenetic state in the nucleus, a way to change what is being transcribed or made in the nucleus via this signaling, distinct from that at the plasma membrane, which also is longer term. 56, 57. Clinical significance, edit. Main article, ciliopathy. Ciliary defects can lead to a number of human diseases. 31, 58. Defects in cilia adversely affect many critical signaling pathways essential to embryonic development and to adult physiology, and thus offer a plausible hypothesis for the often multisymptom nature of diverse ciliopathies. 30, 31. Known ciliopathies include primary ciliary dyskinesia, Bardet-Biedl syndrome, polycystic kidney and liver disease, nephronophthisis, Alstrom syndrome, Meckel-Gruber syndrome, Sensenbrenner syndrome, and some forms of retinal degeneration. 30, 52. Genetic mutations compromising the proper functioning of cilia, ciliopathies, can cause chronic disorders such as primary ciliary dyskinesia, PCD, nephronophthisis, and Senior-Gren syndrome. In addition, a defect of the primary cilium in the renal tubule cells can lead to polycystic kidney disease, PKD. In another, explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Genetic disorder called Bardet-Biedl syndrome, BBS. The mutant gene products are the components in the basal body and cilia. 30. Defects in cilia cells are linked to obesity and often pronounced in type 2 diabetes. Several studies already showed impaired glucose tolerance and reduction in the insulin secretion in the ciliopathy models. Moreover, the number and length of cilia was decreased in the type 2 diabetes models. 59. Epithelial sodium channels, ENaCs, that are expressed along the length of cilia regulate periciliary fluid level. Mutations that decrease the activity of ENaCs result in multisystem pseudohypoaldosteronism, that is associated with fertility problems. 13. In cystic fibrosis that results from mutations in the chloride channel CFTR, ENaC activity is enhanced, leading to a severe reduction of the fluid level that causes complications and infections in the respiratory airways. 41. Since the flagellum of human sperm has the same internal structure of a cilium, ciliary dysfunction can also be responsible for male infertility. 60. There is an association of primary ciliary dyskinesia with left-right anatomic abnormalities such as situs inversus. A combination of findings is known as Cartagena syndrome, and situs ambiguus, also known as heterotaxy syndrome. 61. These left-right anatomic abnormalities can also result in congenital heart disease. 62. It has been shown that proper cilial function is responsible for the normal left-right asymmetry in mammals. 63. The diverse outcomes caused by ciliary dysfunction may result from alleles of different strengths that compromise ciliary functions in different ways or to different extents. Many ciliopathies are inherited in a Mendelian manner, but specific genetic interactions between distinct functional ciliary complexes, such as transition zone and BBS complexes, can alter the phenotypic manifestations of recessive ciliopathies. 64, 65. Some mutations in transition zone proteins can cause specific serious ciliopathies. 66. Extracellular changes, edit. Reduction of cilia function can also result from infection, Research into biofilms has shown that bacteria can alter cilia. A biofilm is a community of bacteria of either the same or multiple species of bacteria. The cluster of cells secretes different factors which form an extracellular matrix. Cilia in the respiratory system is known to move mucus and pathogens out of the airways. It has been found that patients with biofilm-positive infections have impaired cilia function. The impairment may present as decreased motion or reduction in the number of cilia. Though these changes result from an external source, they still affect the pathogenicity of the bacteria, progression of infection, and how it is treated. 67. The transportation of the immature egg cell and the embryo to the uterus for implantation depends on the combination of regulated smooth muscle contractions and ciliary beating. Dysfunction in this transportation can result in an ectopic pregnancy where the embryo is implanted, usually, in the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF fallopian tube before reaching its proper destination of the uterus. Many factors can affect this stage, including infection and menstrual cycle hormones. Smoking, causing inflammation and infection, can reduce the numbers of cilia, and the ciliary beat can be affected by hormonal changes. 1568. Primary cilia in pancreatic cells, edit. The pancreas is a mixture of highly differentiated exocrine and endocrine cells. Primary cilia are present in exocrine cells, which are centrosome duct cells. 69. 32. Endocrine tissue is composed of different hormone-secreting cells. Insulin-secreting beta cells and glucagon-secreting alpha cells are highly ciliated. 70. 71. See also, edit. Molecular machine # biological molecular machines molecular scale artificial or biological device. Protein dynamics # global flexibility, multiple domains study of how proteins move and change shape. Protein domain # domains and protein flexibility self-stable region of a protein's chain that folds independently from the rest. Stereocilia cell structure. References, edit. 1. Definition of cilium. www.merriam-webster.com. Retrieved 15 April 2022. 2. ABCDEF Gardner MB, September 2005. The Importance of Being Cilia, PDF. HHMI Bulletin. 18. 2. Retrieved 26 July 2008. 3. AB Hamo LT, Rosenbaum JL, December 1981. Cilia, flagella, and microtubules. The Journal of Cell Biology. 91, 3 PT 2125S130S2, 10.1083/JCB.91.3.125S. PMC 2112827. PMID 6459327. 4. AB Alberts, Bruce, 2015. Molecular Biology of the Cell, 6 ed. New York, NY. pp. 941942. ISBN 9780815344643. 5. ABC Falk, N. Loz, M. Schroeder, N. Giesel, O. The 11th of September 2015. Specialized cilia in mammalian sensory systems. cells. 4, 3519 do, 10.3390/cells 4030500. PMC 4588048. PMID 263785883. 6 Carat Weekly, DN, September 2021. Primary cilia, turning points in establishing their ubiquity, sensory role and the pathological consequences of dysfunction. Journal of Cell Communication and Signaling. 15, 3291297 do. 10.1007/S12079-021-00615-5. PMC 82222448. PMID 3397046. 7 Carat AB Fish, C. Dupuis Williams, P. June 2011. Ultrastructure of cilia and flagella, Back to the Future. Biology of the Cell. 103, 624970 do. 10.1042/BC20100139. PMID 217289999. S2 SID 76363387. 8 Carat Provo, B. Scalay, J.M., Peterman, E.J.G., September 2017. Intraflagellar Transport, Mechanisms of Motor Action, Cooperation, and Cargo Delivery. The Febs Journal. 284, 18 2905 2931. Do. 10.1111/Febs.14068. PMC 56033355. PMID 283422295. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 9 Carat Elliott, Kelsey H. Brugman, Samantha O. 1 March 2019. Sending Mixed Signals, Cilia-Dependent Signaling During Development and Disease. Developmental Biology. 447, 12841. Do. 10.1016/J.udbio.2018.03.007. ISSN 1095-564X. PMC 61369992. PMID 295489442. 10 Carat ABC Singler, Vena, writer, Jeremy F. 4 August 2006. The primary cilium as the cell's antenna. Signaling at a sensory organelle. Science. 313, 5787 629633. Bibcode, 2006 SCI, 313629S. Do. 10.1126/science.1124534. ISSN 1095-9203. PMID 168888132. S2 SID 29885142. 11 Carat AB Patel, millimeter, Shukas, L. 1 November 2021. Insights into the regulation of ciliary disassembly. Cells. 10, 11 2977 do. 10.3390/cells 10112977. PMC 8616418. PMID 3483120. 12 Carat ABC Horani, A. Furcall, T. May 2018. Advances in the genetics of primary ciliary dyskinesia. Chest. 154, 3645652 do. 10.1016/j.chest.2018.05007. PMC 61303227. PMID 298000551. 13-carat ABCDEF Inuka Y, Hanukolui I, Edelhito, Vanine H, Hanukolui, March 2012. Epithelial sodium channels, ENaC, are uniformly distributed on motile cilia in the oviduct and the respiratory airways. Histochemistry and cell biology. 137, 333953 do. 10.1007/S00418-011-09004-1. PMID 222072444. S2 SID 151789040. 14-carat bloodgood, Ra, the 15th of February 2010. Sensory reception is an attribute of both primary cilia and motile cilia. Journal of Cell Science. 123, PT 45059 do. 10.1042/JCS.066308. PMID 201449998. S2 SID 207165576. 15-carat ABC Panelli, DM, Phillips, CH, Brady, PC, 2015. Incidence, diagnosis and management of tubal and non-tubal ectopic pregnancies, a review. Fertility research and practice. 1, 15. Do. 10.1186/S40738. 015-00008Z. PMC 5424401. PMID 286200520. 16-carat AB Disgrange, A, Legaric, JF, Malik, SM, the 22nd of November 2018. Left-right asymmetry in heart. Development and disease, forming the right loop, PDF. Development. 145, 22. Do. 10.1042/dev.162776. PMID 304671008. S2 SID 537194558. Archived, PDF, from the original. On the 9th of October 2022. 17-carat Rootlin. Retrieved the 28th of March 2022. 18-carat Garcia, Gallo, Raleigh, David R. Writer, Jeremy F. The 23rd of April 2018. How the ciliary membrane is organized inside out to communicate outside in. Current biology. 28, 8R421R434. Do. 10.1016/J.Cub.2018.03.010. ISSN 1879-0445. PMC 64349934. PMID 296892227. 19-carat Garcia Gonzalo, Francesca. Writer, Jeremy F. The 1st of February 2017. Open Sesame, How Transition Fibers and the Transition Zone Control Ciliary Composition. Cold Spring Harbor Perspectives in Biology. 9, 20281334 do. 10.1101/Kshperspect.0281134. ISSN 1943-0264. PMC 5287074. PMID 277700015. 20 Gonçalves, João, Pelletier, Lawrence, April 2017. The Ciliary Transition Zone, Finding the Pieces and Assembling the Gate. Molecules and Cells. 40, 42435253. Do. 10.14348/molchels.2017.0054. ISSN 0219-1032. PMC 5424270. PMID 28401750. 21 Rosenbaum J.L., Whitman G.B., November 2002. Intraflagellar Transport. Nature Reviews. Molecular Cell Biology. 3, 11.81325 do. 10.1038/NRM952. PMID 124152299. S2 SID 12130216. 22 Scalay J.M., January 2008. Intraflagellar transport motors in cilia, moving along the cells. Antenna. The Journal of Cell Biology. 180, 12329 do. 10.1083/JCB.2007091133. PMC 2213603. PMID 1818038. 23 Rohat Jaya, Snell W.J., August 2010. The Ciliary Membrane. Current Opinion in Cell Biology. 22. 454146 do. 10.1016/J.Seb.2010.03.010. PMC 29102237. PMID 203999632. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 24 Wolfram, U, Schmidt, A, June 2000. Rhodopsin transport in the membrane of the connecting cilium of mammalian photoreceptor cells. Cell motility and the cytoskeleton. 46, 295-107. Do. 10.1002/1097-0169, 200,006, 46:23.0.co, 2q. PMID 10891855. 25 Sater, Peter, 2017. Cilia, Before and After. Cilia. 6, 1. Do. 10.13016/S13630-017-0046-8. ISSN 2046-2530. PMC 5343305. PMID 28293419. 26 Wagner CA, 2008. News from the Cyst, Insights into Polycystic Kidney Disease. Journal of Nephrology. 21, 11416. PMID 1826490. Permanent Dead Link. 27 Bruegner M., June 2007. Heterotaxia, Congenital Heart Disease, and Primary Ciliary Dyskinesia. Circulation. 115, 22 2793-95 do. 10.1161/circulation/AHA.107.6999256. PMID 175487739. S2 SID 144058881. 28 2 Mackay, it al. 2019. Primary cilia defects causing mitral valve prolapse. SCI. Tronsa. Med. 11. 493 AX 0290 10.1126/citronsom.AX 0290. PMC 7331025. PMID 311182889. 29 Chen, Holly Y. Kelly, Ryan Lee, Tiunson, Swaroop, and 31 July 2020. Primary cilia biogenesis and associated retinal ciliopathies. Seminars in Cell and Developmental Biology. 110, 7088. Do. 10.1016/j.semct.2020.07.013. ISSN 1096-3634. PMC 7855621. PMID 327479192. 30 ABCD Badeno JL, Mitsuma N, Beals PL, Katsanis N, 2006. The Ciliopathies, an emerging class of human genetic disorders. Annual Review of Genomics and Human Genetics. 7, 12548. Do. 10.1146/anav.genome.7.0805005.115610. PMID 167228803. 31 ABCD Writer, Jeremy F. Leras, Michel R. September 2017. Genes and Molecular Pathways. Underpinning Ciliopathies. Nature Reviews. Molecular Cell Biology. 18, 9533547. Do. 10.1038/nrm.2017.60. ISSN 1471-0080. PMC 5851292. PMID 286985999. 32 AB Hegii, P. Peterson, O. 2013. The Exocrine Pancreas, the Acinoductal Tango in Physiology and Pathophysiology. Reviews of Physiology, Biochemistry and Pharmacology. 165, 130. Do. 10.1007/112_2013_14. ISBN 978-3-319-00998-8. PMID 238813110. 33 Pan J, Snell W, June 2007. The Primary Cilium, Keeper of the Key to Cell Division. Cell. 129, 7. 1255-57. Do. 10.1016/j.cell.2007.06.018. PMID 176047115. S2 SID 1771215. 34 Pugakiva EN, Jablonski SA, Hartman TR, Hensky P, Golemis EA, June 2007. HEF1 dependent. Aurora activation induces disassembly of the primary cilium. Cell. 129, 7 1351-63. Do. 10.1016/j.cell.2007.04.035. PMC 2504417. PMID 176044723. 35 AB Satur P. Christensen St. June 2008. Structure and function of mammalian cilia. Histochemistry and cell biology. 129, 668793 do. 10.1007/00418-008-016-9. PMC 2386530. PMID 183652335. 36 Wong, Sunny Y. Writer, Jeremy F. 2008. The primary cilium at the crossroads of mammalian hedgehog signaling. Current topics in developmental biology. 85, 225260. Do. 10.1016/S0070. 2153, 08, 00809-0. ISSN 0070-2153. PMC 2653622. PMID 191470008. 37 Wagi, Naslamova L., Hancock J.T., 2018. Signaling through the primary cilium. Frontiers in Cell and Developmental Biology. 6, 8. Do. 10.3039/FSL.2018.00008. PMC 58095111. PMID 29473038. 38 Wang, D. Ju, J. 2021. The kinocilia of cochlear hair cells, structures, functions, and diseases. Frontiers in Cell and Developmental Biology. 9, 715037. Do. 10.3339/FSL.2021.7155037. PMC 8374625. PMID 34422834. 39 AB Takeda, Sen, Narita, Kishi, February 2012. Structure and function of vertebrate cilia, towards a new taxonomy. Differentiation. 83, 2S4S11 do. 10.1016/j.difference.2011.11.002. PMID 221189131. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 40 Benjamin Lewin, 2007. Cells. Jones and Bartlett Learning. p. 359. ISBN 978-0-7637-3905-8. 41 AB Hanyukolua, April 2016. Epithelial sodium channel, ENaC, family, phylogeny. Structure function, tissue distribution, and associated inherited diseases. Gene. 579, 295-132. Do. 10.1016/j.gene.2015.12.061. PMC 47565657. PMID 26772908. 42 ABCD Scoen Wolf, Gary C. 2015. Lessons Human Embryology, 5th ed. Philadelphia, Pa. P64. ISBN 9781455706846. 43 Lindemann, C.B., Liesig, K.A., the 15th of February 2010. Flagellar and ciliary beating, the proven and the possible. Journal of Cell Science. 123, pt 451928 do. 10.1242/JCS.051326. PMID 2014500. S2 SID 186735550. 44 Valentine, M. Van Houten, J. the 24th of September 2021. Using paramecium as a model for ciliopathies. Genes. 12, 10 1493 do. 10.3394/genes 12101493. PMC 8535419. PMID 346808887. 45 Johnson K.A., Rosenbaum J.L., December 1992. Polarity of flagellar assembly in Chlamydomonas. The Journal of Cell Biology. 119, 6 1605 11 do. 10.1083/JCB.119.6.1605. PMC 2289744. PMID 1281816. 46 Hau L, Thien M, Brustmasker I, Sivlikoluskale G, Lu Y, Akar S, Provo B, Shaham S, Skale. J.M., June 2011. Intraflagellar transport delivers tubulin isotypes to sensory cilium middle and distal segments. Nature Cell Biology. 13, 7/7/1998 do. 10.1038/NCB 2268. PMC 3129367. PMID 216429882. 47 of cilia and cilinis, more on The Panda's Thumb, archived 17 October 2007 at the Wayback Machine. 48 Ishikawa H, Kubo, Zukitar S, May 2005. ODF2 deficient mother centrioles lack distal /subdistal appendages and the ability to generate primary cilia. Nature Cell Biology. 7, 5517. 24 do. 10.1038/NCB 1251. PMID 158520003. S2 SID 35443570. 49 Lamla S, the 22nd of January 2009. Functional characterization of the centrosomal protein CEP170, Ph.D. Ludwig Maximilians Universität München. 50 Alberts, Bruce, 2002. Molecular Motors. Molecular Biology of the Cell. Garland Science. ISBN missing, page needed. 51 King, SM, the 1st of November 2016. Axonal Dynein Arms. Cold Spring Harbor Perspectives in Biology. 8, 11/028100 do. 10.1001/Kshperspect.0281000. PMC 50888525. PMID 275275889. 52 ABC Adams M, Smith UM, Logan CV, Johnson CA, May 2008. Recent advances in the molecular pathology, cell biology and genetics of ciliopathies. Journal of Medical Genetics. 45, 525767. Do. 10.1136/jmg.2007.0549999. PMID 181786828. 53 Walpert, Louis, Tickle, Cheryl, Martinez Arias, Alfonso, 2015. Principles of Development, 5th ed. Oxford University Press. p. 227. 54 Cilia function as calcium-mediated mechanisms that instruct left-right asymmetry, Science, 5. January, 2023, Vol 379, Issue 6627, pp. 71-78, DOI, 10.1126/science.org 7317. 55 Immotile cilia mechanically sense the direction of fluid flow for left-right determination, Science, 5. January, 2023, Vol 379, Issue 6627, pp. 66-71, DOI, 10.1126/science.org 8148. 56 Tamim, Barbara, 4 September 2022. New discovery, synapse hiding in the mice brain may advance our understanding of neuronal communication. Interestingengineering.com. Retrieved 19 October 2022. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 57 Shu, Shusin, Uparjio Euler, Srigakal, Dupui, Vincent, Pang, Song, Deng, Fia, Wan, Jinksha. Walpita, Deepika, Pasoli, H. Amalia, Hauser, Justin, Sanchez Martinez, Sylvia, Brauchi, Sebastian E. Banola, Samba Shiva, Freeman, Melanie, Zhu, C. Shan, Kirch Hao Sen, Tom, Hess, Harold F. Lavis, Luke Lee, Yulong, Chaumont Dubal, Severine, Clapham, David E. 1 September 2022. A serotonergic axon. Cilium synapse drives nuclear signaling to alter chromatin accessibility. Cell 185, 18 3390. 3407.E18. Do. 10.1016/j.cell.2022.07.026. ISSN 0092-8674. PMC 9789380. PMID 36055200. S2 SID 2519588800. University Press Release. Scientists discover new kind of synapse in neurons' tiny hairs. Howard Hughes Medical Institute via phys.org. Retrieved 19 October 2022. 58 Braun, Daniela A. Hildebrandt, Friedhelm, 1 March 2017. Ciliopathies. Cold Spring Harbor. Perspectives in Biology. 9, 302819 do. 10.1101/perspect.0281191. ISSN 1943-0264. PMC 5334254. PMID 277939668. 59 goods, Chantia M. Christu Savina, Sonia, Zirang, Yang, Moed, Tillo, Morazi, Noah, Carlson Edlund. Patrick, Leibiger, Barbara, Leibiger, Ingo B. Ostenson, Claesgoren, Beals, Philip L. Berggren, Paul. Olof, 2014. Ciliary dysfunction impairs beta cell insulin secretion and promotes development of type 2 diabetes in rodents. Nature Communications. 5308. Bibcode, 2014 Natco, 5.5308G. Do, 10.1038 /ncom 6308. PMID 253742274. S2 SID 41645398. 60 Ichioka K, Cohere N, Okubo K, Nishiyama H, Treio, July 2006. Obstructive azoospermia associated with chronic sinopulmonary infection and situs inversus totalis. Urology. 68, 1204.E57. Do, 10.1016/j.urology.2006.01.072. PMID 168505338. 61 Worsley, Callum, Wiracoddy, Uringer, 1 November 2009. Heterotaxy syndrome. Radiopedia.org. Do, 10.53347 /RID 7420. Retrieved 10 June 2022. 62 Kennedy MP, Omran H, Lee M.W., Del S., Morgan L., Molina P.L., Robinson B.V., Minix S.L., Ulbrich H. Severin T., Ahrens P., Lang L., Morillas H.N., No1 P.G., Zariwala Ma, Knowles Mr., June 2007. Congenital heart disease and other heterotaxic defects in a large cohort of patients with primary ciliary dyskinesia. Circulation. 115, 22 2814 21 Do, 10.1161 /circulation aha.106.649038. PMID 175154666. 63 McGrath J., Bruegner M., August 2003. Cilia are at the heart of vertebrate left-right asymmetry. Current opinion in genetics and development. 13, 438592 Do, 10.1016/S0959-437X, 03-000991-1. PMID 128888012. 64 Lage, Carmen C. Zala, Nora Davis, Erica E. Stoetzil, Corinne, Diaz Font, Anna, Rix. Suzanne, Alfred Hell, Majid, Al-Fadl, Majid, Lewis, Richard Allen, Yad, Wafa, Banin, Yale, April 2008. Hypermorphic mutations in syndromic encephalocele genes are associated with Bardet-Biedl syndrome. Nature Genetics. 40, 44434448 Do, 10.1038/ing.97. ISSN 1546-1718. PMID 183272555. S2 SID 5282929. 65 UE, Laura E. Garcia Gonzalo, Francesca. Bowie, Rachel V. Lee, Chunmia, Kennedy, Julie K. Ashrafi, Cavet, Black, Oliver E. Leras, Michelle R. Writer, Jeremy F. November 2015. Conserved. Genetic interactions between ciliopathy complexes cooperatively support ciliogenesis and ciliary signaling. PLOS Genetics. 11E10056272 do 10.1371/journal.pgen.10056227 ISSN 1553 7404 PMC 4635004 PMID 265400106. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 66 Cavalier Smith, T. May 2022. Ciliary Transition Zone Evolution and the Root of the Eukaryote Tree. Implications for episthoconte origin and classification of kingdoms Protozoa, Planta, and Fungi. Protoplasma. 259, 3487593 do 10.100709-021-01665-7 PMC 9010356 PMID 349400909 67 Farsenberg JH, Su WD, Mustafa, Akbarna, Abuzai WM, December 2016. Biofilms in chronic rhinosinusitis, pathophysiology and therapeutic strategies. World Journal of Otorhinolaryngology. Head and neck surgery. 2.421929 do 10.1016/j.joel.2016.03.002 PMC 5698538 PMID 2920457 68 Horn AW, Critchley Ho, March 2012. Mechanisms of Disease, the Endocrinology of Ectopic Pregnancy. Expert Reviews in Molecular Medicine. 14, E7. do 10.1017/erm.2011.2 PMID 2238070 S2 SID 10039212 69 Canotho, Mercia NS, Parza GJ, Hebrochem, 2004. Orc mouse model of polycystic kidney disease. Reveals essential role of primary cilia in pancreatic tissue organization. Development. 131, 14. 3457 3467. do 10.1242/dev.01189 PMID 152262661 70 Zhang Q, Davenport Jr., Croyle MJ, Haycraft CJ, Yoda BK, 2005. Disruption of IFT results in both exocrine and endocrine abnormalities in the pancreas of TG737, ORC, mutant mice. Laboratory investigations. 85, 14564 do 10.1038/labinvest.3700207 PMID 155802885 71 Yamamoto M, Kadoka K, 1986. Electron microscopic observation of the primary cilia in the pancreatic islets. Archivum histologicum japonicum. 49, 4449457 do 10.1679/AOC.49.449 PMID 3545133 External links, edit. Brief summary of importance of cilia to many organs in human physiology. The Ciliary Proteome web page at Johns Hopkins. Categories: cell movement, eukaryotic cell anatomy, organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.